So we had some emailed in questions that I'm going to start to get us going. And a lot of the questions are, some of the questions are very similar. So David asked a question and Kathleen asked a question that are somewhat similar in terms of combination therapies. So now that you have all these tools in this toolbox and you're combining them, a lot of patients have different questions. So David wanted to know, so for a patient who had had a bone marrow transplant, what are the pros and cons of low-dose Revlimid and Darzalex as maintenance therapy? There was also another question from Lori about maintenance therapy where she said and I know you can't answer individual patient questions, so we understand that. You would have to see them in the clinic, but she has IgM kappa myeloma. She started with RVD that didn't work. She did Darapom Dex, and then is continuing with Darapom Dex as maintenance therapy. So what's the current thinking about maintenance therapy? Maybe we want to just kind of start with that, especially in the context of Isotexab or daratumumab or even selinexor.
I'll take a little stab at this. Maintenance therapy has gone through a number of changes across the years in myeloma. The current quote-unquote standard for maintenance therapy, especially after autologous stem cell transplant, is low-dose lenalidomide maintenance, low-dose Revlimid maintenance. Interestingly enough, there's an ongoing study comparing Revlimid versus Darzalex and Revlimid maintenance. And because every trial that's done using Darzalex is named after a constellation, so we have the Andromeda study, the Castor and Pollux study. This study is called Auriga, and Auriga is ongoing now as an international study to compare Revlimid versus daratumumab and Revlimid maintenance. So we don't know the results just yet of that. There's been a thought for a number of years about using proteasome inhibitors as maintenance. So there was a study called the Tourmaline-3 study, and I love where these names come from. It's called tourmaline because it's using Ninlaro or Ixazomib, which is a proteasome inhibitor, and it has boron at the core, and tourmaline is a boron-containing precious element. And the tourmaline study it showed improvement in progression-free survival, but not overall survival the way that Revlimid did. And perhaps one of the best people to actually expound on this is Dr. Kaufman, because he and his colleagues at Winship have really gone above and beyond to look at optimal maintenance strategies for higher risk patients. And they actually recently had a paper looking at not necessarily just doing one drug that we do for many patients, but looking at combining 2 or even 3 drugs for people with higher risk. So I would really love to hear Dr. Kaufman's thoughts on the matter.
You're correct, is that we manage our standard risk patients differently than our high risk patients in terms of maintenance therapy. For our standard risk patient, our standard continues to be Revlimid as a single agent, and the standard approach is to use Revlimid as a single agent until progression. And just to be very clear, when we're talking about maintenance, we're either talking about therapy after transplant for the transplant patient or therapy after initial therapy for the newly diagnosed patient. For our high-risk patients, we have a long experience, and it was really based on the fact that when we were using single-agent maintenance therapy for high-risk patients, we were extremely frustrated that many patients would relapse early. And without a large phase 3 study, we had a practice of combining combination of a proteasome inhibitor, whether it's Velcade or Kyprolis, and with Revlimid as maintenance therapy. We currently have a study where we're asking the question, is Kyprolis and pomalidomide a good maintenance therapy in high-risk patients? But that continues to be our standard approach. And I think it's a lot of practice's standard approach without large phase 3 studies. There is a very nice study done in Italy where they're asking many questions using a combination of Kyprolis and Revlimid. And we will see that information from the combination of Kyprolis and Revlimid versus Revlimid alone in patients who had transplant or no transplant, and Kyprolis, Revlimid, dexamethasone as part of their induction. And so that study, I think, will be the first time that we'll actually formally answer that question And hopefully there's enough patients in the high-risk subset that we can see a difference if a difference truly exists. I will make just one brief comment about in patients who have relapse disease and then go on a regimen like daratumumab and pomalidomide or Isotexab and pomalidomide. All of those regimens, when they were studied as treatment into progression. And so I at least conceptually, when a patient has relapse disease and I start them on therapy and I continue that therapy indefinitely, I don't really think of that as maintenance therapy. I really think of maintenance therapy as part of the induction therapy, because that's how it was studied. And for the patient who has relapse disease, in general, I don't stop therapy as long as it's working and as long as it's tolerable.
Dr. Richardson, do you want to weigh in?
Yeah, please. I think that the excellent comments from Josh and Jonathan, I think I'll just build on that by saying that just as Jonathan outlined with the combination of lenalidomide and bortezomib in the upfront setting, we've embraced exactly that paradigm of lenalidomide maintenance and at the same time adding a proteasome inhibitor to the platform. And I think that we've got really nice data to support that. And obviously the RVD 1000 experience from Jonathan's center is very informative. I think we also have other studies that support it as well. And so, for example, in our practice, we will integrate a proteasome inhibitor with lenalidomide, especially in high risk. But frankly, if patients choose to keep transplant in reserve, we've actually been embracing the idea of continuing the immunomodulatory drug plus a proteasome inhibitor in those patients, because at the end of the day, high risk versus standard risk, again, myeloma remains a huge challenge and it can change. So I think we've taken the view that sort of a reductionist approach, particularly if the combination is being well tolerated, doesn't necessarily make sense. And that in fact, if a therapy is successful, continuing treatment, adjusting it to improve quality of life and flexibility is very relevant. And I think in that spirit, I was very struck at the EHA meeting by the data of MM004, was presented by Thanos Dimopoulos, which was the use of ixazomib maintenance in non-transplant eligible patients. It was a large trial, mainly ex-US, as you'd imagine, and it was over 700 patients. And Thanos demonstrated very nicely that ixazomib compared to placebo, recognizing that's not a trial that I think any of us would embrace in the US anymore, given that we now know maintenance really matters. When this study was done and designed and done primarily ex-US, there was a rationale for not necessarily offering a maintenance approach to the control group. Going forward, as I say, I would stress that's no longer justified in my opinion. Having said that, the message from this trial was that ixazomib actually doubled progression-free survival to about 18 months from 9 months for the control group. And I think that's very compelling evidence at such a high level that the use of ixazomib as a PI that's oral and so convenient may be quite helpful to patients actually, particularly in the COVID era, where we want to minimize exposures to patients to clinic, and we're using lenalidomide as our backbone and want to add something to it. And my only comment for the patient who mentioned that she has IgM disease and RVD had not served her as well as we would have hoped, and she went on to the daratumumab dexes. I'd absolutely agree with Jonathan's comment that continuous therapy, whether it be part of maintenance or part of relapse salvage, is to me a standard approach to myeloma, because it's a disease that will come back at you otherwise, for sure, in my opinion, and therefore that continuous approach matters. My only comment to her would be to think a little bit about the role, or to certainly be aware that a proteasome inhibitor in her future may be very important, because in my experience, IgM myeloma can be quite challenging. It's a rare subset, but it can be a disease that can be particularly challenging. So just to bear that in mind.
That's so interesting that you say that, because that was one of my doctor's biggest comments for high-risk myeloma. He just said, don't let it get out of control, because if you lose control over it, it's really hard to get back in control, so stay on top of it. I don't know if anybody else has comments about that in terms of high-risk myeloma. And I know, Greg, you had some other emailed questions.
Yes, Margaret asks, I am currently taking daratumumab, Pomalyst, and Dex. What is the protocol for transferring to daratumumab subcutaneous?
And Dr. Kaufman, you covered that really well.
Yeah,
But go ahead.
I mean, I'll just say what we do, and I'd be interested to see if the other panelists have a different approach. With the subsequent cycle of treatment, we go through our standard insurance approval process, and with your next cycle, we just give you the daratumumab, but instead of intravenous, do it subcutaneous. I think a question came up is if you're getting an IV for Benadryl and IV for steroids, do you need to do that IV if you do subcutaneous? And our approach is the answer is no. That on some levels defeats the purpose. And so we give all of our pre-medications with sub-Q, we give the intravenous. And you can imagine all these really convenient ways to do things. You could take your own Benadryl and dexamethasone and, on your way in, call and say, hey, I'm coming in. And then, really do this quickly instead of having it be a half day or an all day event.
I also have a question from Shayla. I'm 38 and work full time and want to start a treatment that has fewer side effects and toxicity. Of the 3 drugs discussed today, which has the least toxicity and most efficacy? I have heard that selinexor has a very high toxicity level and can cause a lot of side effects. Is that true?
Of course, the devil is in the details. So if without knowing what therapy she's had before, it's hard to say exactly which one is appropriate. I think that if you lined up all 3 of these drugs in a vacuum, selinexor does unfortunately have the most toxicity, although it's a pill as opposed to the others, which are given either subcutaneous or intravenously. If she's looking for a drug to be treated on up front, of the 3, the only drug that's approved in the newly diagnosed setting is Darzalex. So it would really depend upon where her disease is at, the finer points of it, and what therapy she's had before.
I would completely agree, Josh, and I would just add a couple of points to that, because I think Jonathan covered the real benefit of sub-Q Dara beautifully. I mean, especially in the COVID era, it's incredibly useful because it cuts down clinic time. I think as we think of isotexab, it's important to share with the audience that one of the differences between ISA and DARA is that there's less complement activation with ISA, which means actually the infusion time has always been short. Now, recognizing that rapid infusion DARA is also readily available now, so the differences are much less sort of profound, they're much tighter. But in that regard also, isotexab has a convenience factor. It's also administered every 2 weeks once one gets established. It's weekly for the first month and then every 2 weeks. It also requires somewhat less in the way of steroid pre-medication because, again, there's less complement activation, which is a potential advantage. But it is not sub-Q yet. And there are actually efforts to go sub-Q in terms of some studies that are currently ongoing to look at that. So we're always thinking about how we can try and make things easier and more patient-friendly. In terms of the patient's question just now, I just completely concur with Josh. It all depends on the context. But if you're working full-time and need the convenience of something that would allow you to do that, I applaud that, number one. But number two, it's very contextual. I think with the antibodies, it's fair to say, in my experience, they're generally well tolerated. I do think a relevant side effect for both ISA and DARA is fatigue. Those are real. But again, if you time the timing of your infusion, for example, and say if you're working and you wish to have your infusion on Friday, that fatigue, in my experience with both antibodies, doesn't last the whole period of a cycle. It lasts a relatively short period. You can then titrate your timing, as it were. So if you had it on a Friday, you might feel better by the Monday or the Tuesday when you next need to work.
Okay, and can I ask you all to expand on this topic, I guess? I don't know, and maybe I'm springing this on everybody, but did you say the Mayo Clinic study or the test that they developed by Dr. Stewart and Dr. Bergsagel, where they were trying to fine-tune how these drugs are used and when they're best used? So this, like her question as a patient is which has the less side effects, but maybe that's not the right question. Like what's the most efficacious for me with the lowest side effects? So like in that Mayo Clinic test, it was talking about how selinexor was seen to be better for higher risk patients and when used later. So this is why we suggest patients go to you because it's so incredibly nuanced. It's so incredibly sophisticated. And now that you have all these tools, how do you determine when you use each of these and in what combination? And just maybe you want to discuss what the field is learning as a whole on this.
This is such an important question in myeloma. And I think the work done by Keith and Leaf is exciting, and I've started to have quite a few patients ask me about it. I don't think it's ready for us to incorporate into our practice, and I'm happy to have people disagree with me. I don't think we're ready for this. I think we're really from a predictive medicine standpoint. We really are. There's only really one drug where we can use an underlying biology and figure out what drug to use. And this is a drug that's not even approved for myeloma. This is using venetoclax and 1114. That right now is the only drug we have where we truly have predictive medicine. So having said that, how do we make decisions about patients? This is, and again, and not trying to make this a black box, but we each of us, we have essentially algorithms, and we base it on the patient's, what the aggressiveness of the disease, the performance status of the patient, what else is going on with the patient. Again, like this patient we talked about here, what are the desires of the patient? What's the risk? Is the patient risk averse and say, I don't want any side effects? Or is the patient say, I'll take any side effect there is, but I just want to get better? All these things we figure out into how to make that decision. And so it's, but the future state where we can know what the right drug is for every patient is where we're all trying to get to.
Dr. Richter and Dr. Richardson, do you want to weigh in on that topic? Because it's a very broad and important topic. Excellent. Thank you, Dr. Kaufman. That's it's why we see you.
Yeah, I think I would completely concur with Jonathan. I mean, these things are so amazing, and we're starting to look at some of this in the earlier phases. I think when we look at patients with standard risk disease and we use some of our standard therapies, we get really, really long outcomes for many of our patients. So there's a variety of platforms like what Dr. Stewart and Bergsegler are doing, and at our institution, Samir Parikh has what's called an RNA-seq whole exome sequencing pathway that we look and we can get some insights into what drives an individual's myeloma forward and specifically target that. But the issue is, in the absence of really knowing how much that driver is going to affect things, we may opt for standard therapies early on and may take those options later on. So when we look at some of these genetic markers, it's hard to know sometimes if these are what are called driver mutations or passerby. And when I say driver, some people's myeloma will find something called a BRAF mutation. And what's unclear in some patients, if that BRAF mutation is driving their myeloma forward, blocking it will improve their myeloma. But in some patients, this may either just be a bystander or maybe what we call subclonal. So maybe only 5 or 10% of the disease has this. So blocking it's not going to control the whole disease. So I pretty much agree with everything that Jonathan says. This is our goal, is to kind of treat myeloma almost like a UTI. You get a UTI, UP, you get a urine culture. A day later, you get this report and says, if you take Bactrim or Cipro, it will go away. That's the goalpost for myeloma.
That'd be great.
Well, yeah, and I echo, obviously, both Jonathan and Joshua in the sense that a goal of a targeted approach, which is tailored to every patient, is absolutely key. And would it be that myeloma is like a UTI? I'm always humbled because at the end of the day, there's some very nice work done with whole genome sequencing that gives us an idea of the complexity of myeloma. And that which I've seen, which humbles me the most, is some of this work, which shows that, for example, one particular patient, this was done, we were able to demonstrate at diagnosis 5,000 mutations in their disease. And after the patient underwent induction remission therapy, transplantation, consolidation, and maintenance, when the patient relapsed, their disease revealed 12,000 mutations. So the mutational thrust of myeloma is in my opinion, unprecedented, and it's probably one of the most important features of the illness. So that being the case, I absolutely agree with Jonathan that I think predictive medicine is a goal, but we're certainly not there yet. And I'm always struck how standard risk can fool me and can actually behave poorly. And I'm also struck that in certain high-risk settings, patients behave remarkably, or their disease, I should say, is much more controllable than we'd imagine. So there's a lovely old saying, and it's true, I think, and I say this very carefully because it's an old saying that a fool with a new tool is still a fool. And by that, I'm not saying any of us are foolish. God help us, no, we're not. But at the same time, we have to recognize the limitations of what we have. So I think we're blessed with multiple options, and I think we have backbone agents that are pleiotropic in their effects. That means they're, for want of a better term, dirty drugs, but they cover multiple targets. And then we have very targeted approaches. And I think Jonathan nailed it when he was talking about venetoclax, a very promising drug. In 11-14, it's a game changer. If you, however, give it to non-11-14, or in those patients in whom one of the important molecular effects, BCL2 expression, is actually low, paradoxically, it can actually make things worse. So there's a complexity here that's really important to understand. So it means that some of our therapies that are targeted, we have to be very careful that we don't use them too broadly, because it may be there are unprecedented or unsuspected challenges that emerge later. And then, frankly, as we think about our current toolbox, we need to be aware of the fact that certain treatments we use at certain stages may buy us a problem later. That's a very important point. So I think Jonathan and Josh have said that very similar things. We are, that's kind of what we're here for. We're here to look at a patient, assess everything, and try and rationalize where we go. And there are certainly patients, for example, in my practice, in whom 2 drugs at relapse may actually suit them very well as a starting point. Then you add a 3rd if you need to. Conversely, there are clearly patients in whom you have to add 3 or 4 drugs right away, because if you don't get a net around the disease and it gets away from you. So I think there's a real art here as well as the science. And until we have watertight predictive medicine tools, I think that art will always remain a key part of what we do.
Okay, and thank you so much for mentioning venetoclax, all of you, because we will be covering that in our next webcast, but that's such an important topic. One of the questions while we're talking about genetics was that a patient had was, what is the impact of the 1Q gain? Are there some drugs that are better off for this particular mutation, or do we just not know yet?
1Q gain, 1Q gain is actually quite complicated. And I'll take the easy part first. We don't have a special treatment yet for 1Q gain. There's laboratory information that where using a drug that targets a particular protein that's similar to the BCL2 protein that's targeted by venetoclax that Paul mentioned, and this other protein is called MCL1, and we have preliminary laboratory data suggests that maybe MCL1 inhibitors will work in the 1Q for 1Q patients. There is a lot of work to do because we don't yet have a really potent and safe MCL1 inhibitor to even try to answer that question. Would MCL1 inhibitors be important in 1Q disease, in 1Q myeloma? Now, 1Q myeloma, there are some patients who have myeloma and they have 1Q abnormalities from the start, and they have multiple abnormalities. Patients who have multiple 1Q abnormalities or multiple copies of 1Q, not just gain of that chromosome once, but gain of that chromosome 2 to several times, that represents a high-risk myeloma, like our classic high-risk myeloma features like deletion 17 and 14, 16, and 414. In addition to being diagnosed and having 1Q, having 1Q rise up later also represents a somewhat more dangerous myeloma. So currently, we don't change our practice a lot about 1Q, but it gives us hints that a myeloma is turning more dangerous.
Okay, Dr. Richardson and Dr. Richter, do you have any comments about 1Q?
Josh first. Yeah, Josh.
In the essence of time, I'm going to take a hard pass because I think Jonathan really summarized everything perfectly.
Yeah, I would just add to that by saying I agree that 1Q is emerging as an important thing. What we do know, though, is that proteasome inhibition in 1Q may matter. It works along the pathway of the MCL1 that Jonathan touched on. And what's very interesting is that actually if you target with an MCL1 inhibitor that actually works very well, interestingly, as a monotherapy, whilst certainly in the most recent work, it's not yet presented, but it's sort of cooking, it's not necessarily been, I mean, there have been no responses, right, Jonathan? That's the challenge we've seen so far with some of the MCL1 inhibitors to date, and there's the cardiac signal. But having said that, proteasome inhibition in 1Q is something I pursue because there is a strong rationale to its use in 1Q and the pathways that are subserved by it. But otherwise, I agree with Josh. I think Jonathan covered it beautifully.
Okay, great. That's great advice. We had a lot of questions from Kate and Carl and Joel also about the difference in daratumumab FastPro versus the IV administration. If daratumumab normally is weight-based and in the dosing, and the sub-Q is just a straight-up dose, is there anything you should consider about that?
When the company who makes the drug made the decision, they very carefully measured how much of the drug was in the body, and we have a visitor. I'll try not to be distracted. They very carefully looked at how much of the drug was in the body and ultimately determined that a fixed dose was able to be enough for the vast majority of patients. And so in preparation for this morning, I really looked, and when they did this large phase 3 study, they really broke it out into the patients who weighed, let's see, 65 kilograms is 143 pounds and less, and 85 is about right at 200 pounds. Paul could help me out a little bit here.
I always lie about my weight. Yeah.
No. The imperial system, if you will.
No, I get it. Yeah.
And there are some small differences in side effects, and there was a question that I answered on the Q&A earlier. There's small differences in the amount of neutropenia, low white blood cell count, for the smaller patients, the patients who weighed less than 65 kilograms, but there wasn't really anything really clinically significant. And so, so no matter what the weight, I haven't changed. Now, then the question becomes, well, what if somebody is, I don't know, 350 pounds, or what if somebody weighs 80 pounds? Should we make a difference? And I don't think we should. I think that it's unlikely to make a major difference.
Okay, and then Bonnie, well, we had some other questions too about renal instances. Is there any difference between IV versus sub-Q with renal function?
There's not.
Okay. Okay, great.
One thing I would just add about the IV versus the sub-Q is that, this drug works very different than a lot of other drugs that we think about. So for many drugs we give, both classical chemotherapy and even some of our novel drugs, there's a clear dose response curve that if you give more drug, it kills more and may cause more toxicity, and up, and up, and so on and so forth. With monoclonals, one of the ways that we kind of measure how much we need is something called receptor occupancy. So think about it, that every cancer cell has a lock and the drug is the key. And at a certain point, every lock has a key in it, and dumping more keys into the system, there are not necessarily more locks. So for a lot of the monoclonal trials, once we get to a certain amount of drug, giving more of it doesn't necessarily change anything.
Excellent point.
Yeah, I agree. I agree, Josh. That's very well put. We have an excellent antibody that, in my opinion, somewhat underappreciated, elotuzumab. And you're absolutely right. We did the studies to show, and Jonathan was very much involved with this, to show that receptor occupancy at 10 was the same as at 20, and therefore the 10 milligram dose per kilo was taken forward. Interestingly, when we give elotuzumab monthly, though, we make it 20 mg per kg as opposed to 10 for the repeated dosings. I think what's so interesting, though, is to share with our patients that remember that with both DARA and ISA, if for any reason as the schedule extends with maintenance approaches, once a good response is seen, we've definitely seen that sometimes disease control can wobble a little bit. And in that situation, it's actually sometimes very valuable to restore frequency of administration of the monoclonal to restore response. And that can be a relatively simple approach that can buy more time in the setting of potentially the monoclonal antibody running out of gas. The only other comment I'd make is when I think of bortezomib sub-Q and bortezomib IV, one area which I think is sometimes underappreciated is that for certain disease settings, it may be valuable to exploit what we call a Cmax. And for example, in extramedullary disease, that's becoming more and more of a challenge for some patients as the natural history of the myeloma extends in each patient. Sometimes a switch from IV to sub-Q in those scenarios can be valuable. But I absolutely agree with you, Josh. There's no evidence that that's the case with the antibodies because it's a different mechanism.
And another dosing question about selinexor. Carol was asking, is it typically given at 80 milligram on day 1 and 3, or can it be given once a week? Is there any difference in the impact if given once a week?
So yeah, so the original study that was done, the STORM studies, which is an acronym standing for selinexor in the treatment of relapsed myeloma, the starting dose was that twice weekly at 80. And in the study, there were several ways that we could dose reduce. So you can either bring it from 80 twice a week down to 60 twice a week, or go from 80 twice a week to 100 milligrams once a week. And in those patients that had very aggressive disease, we found that we needed to give them a higher total dose that was doing all of the heavy lifting. So we had to give the 80 twice a week in order to get to the response rates we needed. When we combine it with other drugs, where it's not doing all of the heavy lifting, it partners and synergizes with drugs like Velcade or pomalidomide, we're able to give it at the once a week dose and still get the responses we need. So overall, I think the majority of the usage of selinexor in the clinic right now, despite the fact it doesn't quite yet have the FDA approval, has been in the triplet combination. This way you can back off to the once a week dose, still get the efficacy you need, but minimize toxicity.
Yeah. I would like to echo that, Josh. At our center, we've been very pleased with the performance of selinexor once a week in combination with proteasome inhibition, be it either bortezomib or carfilzomib. And I absolutely agree. It's a very different toxicity profile with the once a week schedule. And I thought you said it very nicely in your presentation, Josh, that one of the most exciting aspects of the Boston trial was that this was a real world study. I mean, I think it's important to share that with our audience, that Boston was done across the globe. We had centers participating in Eastern Europe and India, and they were very, unexposed, as it were, to antiemetic strategies. And in fact, in the study, it was only required for Zofran. That was the only requirement. They weren't talking about Zyprexa and some of the other pearls that Josh shared. And even with those limitations, the tolerability profile in the triplet was much, much, much better than had been seen previously. And what was also very interesting, and Josh mentioned it, was I think it was a very brave study because it put bortezomib weekly against the classic schedule, still preserved efficacy, and then remarkably reduced neurotoxicity. Now, that may be more than just the weekly sub-Q bortezomib, and we're learning a lot about selinexor, and it has anti-inflammatory properties that may actually be relevant to the more severe neuropathies that we see. Patients, I think, if I may, Jen, need to sort of hear this, that neuropathy is very complicated. It's part of both the disease and also inflammation. And if you can modulate both neuropathy, the severity of neuropathy can be reduced. So hence, that's why we tend to be very picky about partnering with bortezomib steroid on the day of it and the day after, because there's clear evidence that that's a better way to go. In that same spirit, the partnership of selinexor with bortezomib in the Boston study struck me in that regard, that the actual rates of grade 3 neuropathy were significantly lower. And you might say, well, that's just weekly, isn't it? And possibly it is, but I think the signal actually suggests more than that. And obviously we'll learn more.
Yeah, that's a really good point, a very, very important point. And you're learning all these nuances as you gain more experience in the clinic, and there's just so much to learn. Someone else, Patrice, asked, have any of these drugs been used in combination with bellamath? And what are your thoughts of how bellamath compares to some of these or in combination with some of these? And somebody might want to explain what bellamath is.
Bellamath is a monoclonal antibody against a new target called BCMA, B cell maturation antigen. The good thing about BCMA as a target is that it's essentially solely on plasma cells, and myeloma, a cancer of plasma cells. You really can target that protein on these cells and not have, from a target perspective, not have a lot of off-target impact. Now, bellamath is an antibody, and then combined to the antibody is a chemotherapy, more of a conventional chemotherapy. So in theory, the antibody attaches to the myeloma cell, gets brought into the myeloma cell, and delivers the chemotherapy directly to the myeloma cell. So this drug has been studied primarily so far as a single agent, but just almost like all medications in myeloma, it starts out as a single agent and then starts to have additional combinations. So the largest studies to date as a single agent shows that it works in about a third of patients, which is similar to what we saw when daratumumab was used as a single agent, similar to what we saw with bortezomib and lenalidomide as a single agent, and or in combination with dexamethasone. So as a single agent, it's in that range of what we think is going to be effective therapy. Just to not get too far off, and I'll get to the question. There is toxicity with the drug. The major toxicity has to do with ocular problems and inflammation and vision difficulty. So hopefully it'll get approved soon, but it'll be very important that we understand those potential side effects. From a combination standpoint, my colleague here at Emory, Ajay Nuka, just presented data of the combination with Velcade. It looks to be safe and effective. There's combinations with Revlimid or lenalidomide, which hopefully will show us good data. We have yet, if these are ongoing, I don't know about it, and that's okay. I don't think that there's going to be a big push to combine monoclonal antibodies. I don't think we've been terribly successful with that approach yet. Certainly could be combined with selinexor because they have really different side effect profiles. The study right now, there's a study right now of looking at what is the best combination for bellamath, and I'd be interested to hear what my co-panelists think. What could be that best combination?
Josh?
So all of the combination studies, or all the studies with bellamath, have the word DREAM. So it's DREAM 1, 2, 3, 4, 5, and the single agent is a DREAM 2, and the bortezomib is DREAM 6, and they're combining with everything. I think it's a little soon to know what the optimal combination is going to be. One of the first hurdles is actually what's called the ODAC is meeting next week, I believe. So for those who may not know, when oncology drugs, oftentimes before they go to the FDA, have a little stopover. It's something called the ODAC, the Oncology Drug Advisory Committee. And they have experts who have used the drug in clinical trial, along with what's perhaps the most important part, patient advocates come to share their experience on the clinical trial to explain to the FDA what the true risks and benefits are at the bedside. And what was released, I believe yesterday, was actually the list of some of the things that the ODAC wants to know, which is exactly as Jonathan pointed out. One of the big concerns is ocular toxicity. And so far, many of our strategies haven't quite prevented that. We've tried things like steroid eye drops. So very hopeful that the drug is going to be approved shortly, but there's a few small hurdles the drug still has to overcome.
Yes, thank you, Josh. I think that's very well said. Full disclosure, I'm actually participating in the ODAC on Tuesday with two of my patients, actually, and I'm extremely grateful to them because they both participated in Dream 1 and Dream 2 and have done remarkably well. I think the important message is exactly that the keratopathy we've come to understand much more in detail. Jonathan made the point that there may be some inflammation. We've actually learned that perhaps it's less than we initially thought, and that's why the steroid drops were less helpful than we'd hoped for. But what we have realized and learned is that dose and schedule really matters. And if you drop the dose and increase the interval, the keratopathy is much less of a problem. And I would also stress that the keratopathy we've seen, we've been extremely careful to document and grade it very conservatively. So, patients may see a lot of quote unquote grade 3 keratopathy. What you have to appreciate as you see that is a grade 3 keratopathy as defined by the grading system used in the trials was very conservative, appropriately, because vision is very important. But having said that, all of my patients, none discontinued for reasons of keratopathy. And whilst it is a challenge and we're working on it, the important message is that generally speaking, it's mild to moderate and it's reversible. And that's very important. To the point of combinations, therein I think lies the answer, because if you can extend the dose and interval, extend the interval and reduce the dose, keratopathy is simply not a major problem. And so combinatorial strategies will really matter. So I agree with Jonathan and Josh that there are a whole variety of combinatorial strategies being pursued, and all of them, in my view, from what I've seen, are looking good. We're actually leading the Dream 5 trial, where actually, Jonathan, we're bringing in actually an antibody into two arms of the study, different ones, that augment or optimize T cell function. So the idea is that we use the immunogenic cell death that belantamab mafodotin generates targeting BCMA, and we bring in a T cell targeting antibody that further augments that. And obviously safety is key. The other part of the Dream 5 study is actually a gamma secretase inhibitor. Gamma secretases actually have an effect on BCMA expression. They down-regulate it. So the idea of this simple pill, which is very safe, and it was actually originally studied interestingly in Alzheimer's disease and was quite safe, just unfortunately wasn't as effective in Alzheimer's as we would have liked. But in any event, we're repurposing it, and it's going into the space with belantamab to try and augment activities. So, Jenny, the message for patients is a lot going on. And what I particularly like, frankly, about the antibody drug conjugate platform is it's an every 3 to 4 week infusion. You come into the clinic, you get it over a couple of hours. And by the way, you don't have to give steroids with it because it doesn't need them. So this is for the real world that we're dealing with now, post-COVID or in the middle of COVID, probably a better way of putting it. This is, I think, a very important platform. And I share your view, Josh. I'm very hopeful, and Jonathan, very hopeful that the FDA discussion on Tuesday will be constructive. I do want to share the FDA have been incredible about this. In fact, correct me if I'm wrong, gentlemen, but there hasn't been one ODAC in myeloma in which the FDA have not actually ultimately approved the drug. I think that's correct. Correct me if I'm wrong, but I don't believe there's been one in which they've actually disapproved the drug. At the FDA level, the panel may have had reservations, but the FDA have actually gone on to approve the drug. I think that's true.
Yeah, for the two ODACs that had concerns, ultimately the FDA felt that this should be in the hands of the clinicians and the patients to make the discussion available.
Excellent. Yeah, so that was selinexor and panabinostat, and also the carfilzomib ODAC, which obviously was done many years ago. But the concern there was the cardiac toxicity, and obviously we've come to recognize how to handle it. It's real, it's definite, but we've come to realize how to handle it and how to manage it. Yeah.
Okay, great. I'm going to ask 2 more questions. We have over 50 questions, so I apologize because we're not going to get to all 50 questions. But I'll ask these 2, and then we will maybe invite you to provide some written answers that we'll share back to specific patients who are calling in. But Linda had a question about selinexor. If you failed on Velcade, would you take it with selinexor? And because it's being used in the selinexor Velcade Dex trial, is that trial still open? And that was the question about selinexor.
Sure. So the selinexor Velcade Dex trial is still open, but to my recollection, no longer accruing because I believe it's accrued its
That's right.
entire complement. Again, please correct me if I'm wrong on this.
Yeah, that's correct, Josh. That's correct.
Um, because the study that say was comparing people between the doublet of Velcade and dexamethasone versus the triplet of selinexor Velcade and dexamethasone, patients had to be eligible, or better said, Velcade dexamethasone had to be an appropriate choice for them to go onto the trial. So you couldn't be completely refractory to Velcade and dexamethasone, because if you would have gotten randomized to that group, that would not have been an appropriate choice. So I don't know that we have a deep insight yet into what the response rates would be of the triplet of selinexor Velcade dex in someone who's progressed on Velcade and dexamethasone. There may be a more optimal partner with selinexor for that type of patient. With some of our other drugs, we do know that we can resensitize. So one of the drugs that Dr. Richardson had mentioned, panabinostat, for example, we know that drugs like that can resensitize to proteasome inhibitors or immunomodulatory agents. And there's probably a little more robust data on people progressing on Revdex or bortezomib dex and adding the panabinostat to recapture disease. I'm not as familiar with that data for selinexor, but I open it to the group.
Yeah, no, I'd love to, echo that, Josh. And I think the panabinostat story is fascinating because there is also very nice evidence now from groups in Germany about the upregulation of CD38 by panabinostat, such that there's now concerted efforts to look at, at that effect in terms of monoclonal antibodies. And very importantly, Pano is, we've documented its activity with proteasome inhibition, but Josh touches on an important point. It's underappreciated how active panabinostat is in combination with immunomodulatory drugs, and which Jonathan and I know from our studies, not only with Pano, but other HDACs, which unfortunately are not going forward in development in myeloma, but obviously Pano is fully approved. And I think it shows considerable promise. I think the other thing to share in terms of the Seli-Vel story is that I completely agree with you, Josh. We don't have data for truly bortezomib refractory patients benefiting from the addition of Seli. In that situation, I might suggest that you think about carfilzomib Seli as an obvious next step. But there's lovely data on Seli plus POM presented by Christina Chen at ASH last year from the STOMP study, which was both well tolerated and active. And I think in that sense, there's always been a rationale in our myeloma world that just because one drug has failed a patient, when you combine it with another drug doesn't mean it's not going to work. That's very different to epithelial cancers, for example. In colorectal cancer, if a particular class of drug fails a patient, trying to salvage with the same class doesn't work. That's not true in myeloma, as long as there's a rational combination platform. One point I want to make, Jennifer, in that spirit, is as I think about CD38 antibodies, in, for example, lymphoma, you use Rituxan and B-cell lymphoma, large B-cell lymphoma throughout. So you might do R-CHOP, RICE, REPOC. These are acronyms for chemotherapy regimens for those patients who may not know what that means, but they're cocktails of chemotherapy. The point is Rituxan is used throughout. In my opinion, myeloma is a little more clever than that. And I think that's why, whilst daratumumab is really doing an exceptionally good job up front and is now part of maintenance, I do believe there may be a strong rationale for isotexab, still targeting CD38, but coming into a different space with subtly different properties in the right combination. And I think that's a nice way to think about it because, again, CD38 targeting to me is a backbone of what we do. The question is how most intelligently to do that.
Well, you led into my last question. And that was from.
I want to make sure Jonathan has. Jonathan needs a shout here too.
So my last question is from Mindy, and then Bonnie kind of has a nuanced question around this. So if you fail on daratumumab, can you take isotexab? If you fail on isotexab, can you take daratumumab? We had a show where you interestingly said, well, I'll let you answer that live and not repeat your answer. But Bonnie also said, do you think isotexab may be better than using DARA again at first relapse in a patient who used DARA as part of upfront therapy and only achieved PR or partial response, and then went on to have a stem cell transplant and rev maintenance? So when do you use these, and how? I know daratumumab is earlier probably in development of getting approved for newly diagnosed or upfront in the upfront setting, and isotexab I'm sure will move its way up there. But what's the answer?
The real answer is that we don't. These are great questions, and every myeloma doctor in the world is asking those exact same questions. And the answer is we don't know. I think Paul's mentioned some very some rationale why one why one might be one to use one versus another. I think the answer is we don't know. We don't know. There is no large data set, clinical data set, that helps us figure out if DARA worked previously, would it work in the future? We don't. There's not a reason it wouldn't, but we don't. It hasn't been studied. If somebody had a not great response to DARA up front, some combination, what would be the best monoclonal antibody? Is it trying DARA again, using Isotexab? Should we change targets and use elotuzumab? These are all really great questions. I think, and I think Paul really said it well. In terms of if somebody really was refractory to daratumumab, I'm not sure if my next treatment would be an Isotexab-based treatment, but I think down the line, if I was in a situation where I needed a combination, I probably wouldn't go back to daratumumab. I'd probably use Isotexab in that situation. So, but it's a great question, and we're all going to be trying to answer that question over the next 5 years.
Dr. Richter, do you have an opinion on that as well? And then Dr. Richardson we'd love to hear your opinion too.
The only so I completely agree with Jonathan. The only there is no large data set. There were a handful of people in one of the earlier studies with isoteximab, a study that doesn't have a cool name. It goes by TED 14154, which is not a very easy name. And there was a handful of patients who got single agent isoteximab after single agent daratumumab, and we did see some responses, but what's really important that Jonathan brought up was, I don't know yet that if you were on daratumumab and Revlimid, that my next regimen would be isoteximab, exactly how Paul said it as well. Switching partners and or giving it at a later time is probably the optimal strategy there. But as always, Paul puts it best, so I'd put it to him.
No, Josh, you're too kind. Not at all. I want to applaud Josh on his acronyms. I think it's so interesting, and it's
Very
actually
impressive.
well said.
Very
because they really do mean something. People think we're kind of crazy, but there's endurance, endeavor, determination. All of these studies have names for reasons, and good on you, Josh, for that. No, I so agree with John and Josh about this. I think what's so interesting about the construct, though, is that if you think of the chemo data, you have to be very careful about what we call cross-trial comparisons. And this is, I hope helpful for patients to hear, because there's often a tendency to say, well, a drug behaved like this in one study and it looked a heck of a lot better than the similar class in another. You've got to be very careful about that. But the chemo data were quite striking. And I think they did, they pleasantly reinforced what we learned in Icaria. And I think it does help us think about this, because the one thing is about this apoptotic signal, and I hate to sort of perseverate on it, because as Jonathan is right, it's a hypothesis at the moment, but it is interesting to me that isoteximab and carfilzomib, which I consider the most potent proteasome inhibitor we have by far, the fact is that the performance in Akema was really quite good. And I think that was quite fascinating. And I think as we think about DARA being upfront and as part of maintenance strategies, and they say a patient's been on an IMiD for a prolonged period of time, and then IMiD fails them. By the way, Jen, one little thing. I always like to say this, and I don't want to sound silly, but I always think that drugs fail patients, not the other way around. Our patients don't fail the drugs fail the patient. So just a sort of bet. But anyway, the fact is that when Len fails the patient in that situation, I might say now, based on Akema, you know what? Isoteximab carfils. That might be a really powerful platform, as long as the clinical scenario justifies it. I mean, obviously, if the patient doesn't have cardiovascular risk, etc., I might think about that based upon what I've seen from Akema. But Jonathan said it beautifully. We obviously need clinical information, because we can be so surprised by phase 3 information The best example being Bellini, where that was supposedly a slam dunk in favor of venetoclax, and boy, we learned some lessons out of it. So Jonathan's absolutely right on that tack. But I would say, based upon what we learned from EHA and IKEMA, that might be how I think about the question that's being asked.